Hi everyone. Thanks for visiting my poster. I'm Seong, a graduate student at the Biocyphers Lab at the University of Pennsylvania. My project is about a unified view of transcriptome complexity by combining transcriptome annotation with long and short read RNA sequencing. The main goal of the project is to better understand the isoforms and splicing changes in a given biological condition, and to do so, short read RNA sequencing and long read RNA sequencing are widely used. Short-read RNA sequencing has given researchers unprecedented detection and quantification capabilities of splicing variations across biological condition and disease stage. However, short-read technology is not able to directly identify which isoforms are responsible for the sequence fragments or how splice variations across the gene are connected. In contrast, long-read sequencing offers improved detection of underlying forward partial isoforms, but it's limited by higher range and throughput, which hinders the ability to fully detect and quantify splicing variations. So the question is, can we compare and contrast short-read and long-read RNA sequencing together to combine their advantage while complementing their disadvantage? Here, we compare and contrast the results of both short-read and long-read sequencing together with the annotation of known isoforms to gain further insight of using different sources of information. We use human cell line datasets sequenced by short-read and long-read from the Long-Read RNA-seq Genome Annotation Assessment Project Consortium. We feed short-read RNA-seq with genome annotation into MAGIC to visualize splicing variations and possible isoforms of the gene we're interested in, and feed long-read RNA-seq with genome annotation into long-read algorithm, FLAIR, to get the list of full isoform in GTIFF file format. Long-read algorithm can be different software packages, and the dataset can be sequenced by different technologies such as Oxford Nanopore. And then we analyze the gene splice graph in a modular manner. A module represents distinct segments of a gene splice graph involving overlapping splice junctions which are contained between a single source and single target exon, like in the black dotted box. As the gene can be complex, having multiple splice variations, we want to focus on the subunion of the gene module with a divide and conquer approach. With the modules, we can see which splice junctions are supported by which information. They can be from short read, long read, annotation, or any combination of them. Each splice junction in each module can fall into one of 6 categories. All represents when the splice junction is detected in all sources of information. De novo means when the splice junction is not found from annotation. Therefore, post de novo means the splice junction is found in both MAGIC and FLAIR, but not in annotation. These two rows are detected by just MAGIC or MAGIC and annotation. Given the high accuracy of the short reads, we expect the vast majority should be real, and these are things missed by long reads. FLAIR annotation is the case possibly true but not detected by MAGIC. For FLAIR de novo, given the higher range and low coverage of long reads, these are most expected to include high rate of false positive. In our dataset, we found 106 270 splice junctions in 56 127 modules in 59 56 genes. We sum up all the number of splice junctions in each category in all modules and found that about 50% are supported by all sources of information. Only a small portion, about 1.5%, are supported by both long read and short read. In contrast, over 34% are detected by MAGIC in short reads, of which 20% are supported by annotation, and over 50% are de novo. Given the high accuracy of the short read, we expect the vast majority of those to be true positive. Only 6.6% are detected by long read and then annotation, and over 8% are only supported by a long read that potentially could have high rate of false positive. Now we turn to break down each of the de novo categories to subcomponents to gain further insights. For MAGIC de novo, it can be divided into 2 subcategories, one with combination junction and another with novel junction. Combination junction represents the dark green junction in the upper splice graph showing a new junction that makes a new combination of transcript, and novel junction represents the light green junction in the bottom splice graph showing a novel junction that is not found in annotations such as alternative 3'N, alternative Y''N, or intern retention. For FLAIR de novo, since FLAIR reports isoform as a final output, we break down it into combination junction, novel junction, partial exon, or any combination of those categories. We show FLAIR de novo categories by comparing with MAGIC splice graph. The first row represents splice graph visualized by MAGIC showing 3 exons, 2 junctions found by MAGIC in annotation. The arrows in exon number 1 means transcription start size, and the carrots in exon number 3 means transcription end site. Similarly with MAGIC de novo, FLAIR combination is the case where a new splice junction gives a combination of new transcript which fall into Roman number 1. And flare novel is the case when a novel splice junction is found in flare but not in MAGIC and annotation, which falls into Roman number 2. We also define partial exon. Partial exon means the start or the end of the exon of the transcript is different from the transcription start site or transcription end site. Therefore, partial exon can be only in the first exon or the last exon. Roman number 3 shows when there is both flare, novel, and partial exon. Roman number 4 shows when there is both combination and partial exon. Similarly, Roman number 5 falls into the case when there is both combination and novel, and Roman number 6 is when there are all three of them. Our algorithm is based on splice sites matching, not exons. That being said, Roman number 7, partial exon exists as part of MAGIC, annotation, or a combination of these two, which means there cannot be pure partial exons. We compare and contrast the results of both short read through MAGIC and long read through flare together with the annotation of known isoform to gain further insights of using different sources of information. Our analysis from human cell line data indicates that 35% of the junctions were only detected by short read, and among those, 20% of them were detected by annotation and 50% of them were de novo, while approximately 8% is only from long reads and 6% is from both long reads and annotation. These results point to the potential in combining different sources of information. This is a subset of our results. If you want to hear more of our analysis, I encourage you to attend my poster, C-28. Also, I encourage you to attend another poster about RNA splicing analysis using heterogeneous and large RNA-seq datasets presented by Paul, who is one of our team members in the Biocyphers lab, in poster number C-27. With that, I would like to thank to the members of the Biocyphers lab for all the help and support for the project, and I'm happy to talk more afterward in the poster session. Thank you.